The amoeboid form is widespread among the eukaryotes. Not only amoebas, but also the cells of a huge variety of protists, as well as many cell types of multicellular organisms are amoeboid. Check out an example of a human macrophage cell. It looks like a typical amoeba. But what does it mean? The amoeboid cells are different from the other forms of cells with non-stable, easily changing cell shape. The dynamic outgrowths of the cell surface performing locomotory or feeding functions are called pseudopodia. Let's quickly review the diversity of amoeboid protists. First of all, of course, these are so-called naked labose amoebae. The cell membrane in these organisms, of course, surrounded by glycocalyx, which is typical for all the cells, but otherwise it is usually naked. The labose pseudopodia or lobopodia are thick and blunt and can be single in case of monopodial forms or multiple polypodial forms. The slime molds belonging to the taxon Eumycetozoa are also amoebae, and part of their life cycle is represented just with tiny microscopic amoeboid cells. But they are also capable of forming huge multicellular aggregation, pseudoplasmodia, or giant multinucleated cells, the true plasmodia. And finally, to form fruiting bodies, just like fungi do. Heterolabosian amoebae also form pseudopodia, which are thick, blunt, but its formation is not gradual, but rather fast and spontaneous. This saltatory process is called eruptive movement, the type of amoeboid movement characterizing heterolabosians. There are also amoeboid cells belonging to the group called endomyxa. These cells also change its shape, but in this case pseudopodia are long and thin. In this case they are called phylopodia. Mostly benthic marine protists called foraminiferans are also characterized with thin pseudopodia. These pseudopodia form a network which it uses for feeding and locomotion. Multiple granules are moving along this network, which mostly represent food particles transported along this pseudopodial net. This type of pseudopodia is called granular reticulopodia. The other prominent feature of this group is external shell enclosing the cell body. Radiolarians, the other amoeboid group, are marine and planktonic, and unlike foraminifera, have internal mineral skeletons composed of spicules or even solid internal perforated skeletons having a variety of shapes. And the last type of amoeboids, the heliazoans and also radiolarians, share the similar type of pseudopodia, the exopodia. Exopodia are straight, thin, and supported with a bundle of microtubules. But heliazoans, unlike radiolarians, do not have internal skeleton. Instead, they often have extracellular skeletal elements surrounding the organisms. In conclusion, it is necessary to emphasize that amoeboid protists do not represent a particular evolutionary lineage. Amoeboid cells can be found almost in all macroclusters across all the eukaryotic tree, and the term amoeboid just designates a particular type of cell organization, not a taxon of closely related organisms.